Hello, welcome everyone. We are thrilled to have you here for this webinar today. So over the next 45 minutes, we will delve into the world of drug delivery with a special focus on in vivo delivery and more specifically on the innovative tools from our partner Certest Pharma that facilitate this process. Despite numerous technologies advances, in vivo drug delivery still faces major challenges from ensuring drug stability and precise targeting to overcoming biological barriers and immune responses. So in this session, we invite you to discover how Certest Pharma's exclusive lipids and specialized tools can help overcome these key challenges, enabling more efficient in vivo drug delivery. We hope these presentations provides valuable insights and inspires new possibilities for your research. So let's get started. First, let me introduce the experts joining us today. Dr. Javier Jimenez Warren, our speaker and Chief Technical Officer at Certest Pharma. And during the Q&A session, he will be joined by Dr. Erika Shiri, one of our lab experts who works on RNA production and delivery projects. And I'm Diana Perrier, your host and moderator for this session. Before we begin, here's a quick overview of today's agenda. We will start with some session guidelines, followed by a brief introduction to Tebu Bio, and of course, an introduction to our speaker. Dr. Javier Jimenez Warren will then present Certest Pharma's lipids and solutions designed to enhance in vivo drug delivery and overcome its major challenges. Stay with us until the end for the Q&A session, where our experts will be happy to answer all your questions. I'd like to go over a few housekeeping guidelines for this session. First, this session is being recorded and we will share the recording with all the participants after the webinar. If you have any questions during the presentation, please use the Q&A box located at the bottom of your screen. We will address them during the dedicated Q&A session at the end. And please take a moment to complete the survey that will appear in your browser when you leave the session. Your feedback is invaluable, so we would like to, if you like more information, let us know. We would love to follow up with you. And now a quick word about Tebu Bio. We are a pan-European company, and for over 70 years, our mission has been to support life science researchers by facilitating their projects through our unique positioning and comprehensive range of solutions. Our European coverage ensures easy access to a broad selection of innovative solutions sourced exclusively from trusted and ethical suppliers. If you're short on time or need specialized expertise, you can outsource part or all of your research project to our contract research services lab in Europe. Our team covers everything from mRNA production and formulation to in vivo models development and target identification. We also streamline ordering and logistics through our efficient e-procurement system, backed by decades of experience. To sum it up, with Tebu Bio, you focus on your research and we take care of the rest. Our laboratory has been growing for over 20 years and composed of scientific experts across various fields and a team of versatile technicians. We offer a wide range of services. Our expertise is structured around 4 key platforms: RNA-based therapeutic discovery, supporting RNA production, delivery, and expression; in vitro modeling, culturing and optimizing your experimental models; biomarker mapping and data analysis, providing comprehensive insights into your research. Before introducing our speaker, I'd like to highlight our partnership with Certest Pharma. This collaboration allows us to support you throughout the entire process of developing and delivering an RNA-based drug. From discovery, as covered in our previous webinar in February on RNA synthesis capabilities and formulation screening, to in vivo applications, leveraging the innovative tools provided by our partner Certest Pharma. Without further delay, I'm honored to introduce our speaker for today's webinar, Dr. Javier Jimenez Warren. With a PhD in organic chemistry and over 15 years of experience in synthetic chemistry applied to biotechnology, he has contributed to the development of oncology therapies and innovative vaccine models. As a Chief Technical Officer at Certest Pharma, he leads the lipid department and has filed for patents on lipid families used in lipid nanoparticles since 2021. Additionally, he collaborates with the European Pharmacopoeia, contributing to the drafting of monographs for RNA-based vaccines. With that, I'll hand it over to you, Dr. Jimenez, and thank you again for joining us, and we hope you enjoy this session.
Okay, thanks a lot for that, Diana. Thanks for the presentation. I'm really glad to be here. We're really excited about this partnership that we have between us. It's going to give us the possibility of reaching more people so they can try our technology and see that they can be as happy with the technology as we are. So just to give you a brief idea of what we do in Certest Pharma. So basically, we started in 2021. We started this part of the business, which is related to RNA therapeutics. And in this, we're working in developing new ionizable lipids, also in optimizing the formulations and making them thermostable, as you will see in a minute. We are also very engaged in trying to reach a better targeting of different organs so we can make more efficient therapies. And also, since we have to use RNA in our therapies and to validate our technology, we can also do very good RNAs. We have some UTR and current optimization techniques that we have filed patents for. And we have also a part of the business which is dedicated to oligonucleotides, so we can make the necessary modifications to make oligonucleotides that can reach the market. So I should be able to pass it. Okay, so I'm going to start with the ionizable lipids. So basically, when we started doing ionizable lipids, one of the things that anybody that starts doing ionizable lipids finds is that it's kind of difficult to find a space, structural space that is not covered or it hasn't been covered before. So we encountered that problem, and we had to go with a methodology that a lot of people are using, but which is basically using combinatorial chemistry. But also we made a twist because we included machine learning in our screening process. So basically, we identified a family of ionizable lipids that we thought that might give a good result, and we started the synthesis with combinatorial chemistry. Once we designed and synthesized these lipids, what we did was formulation into LNPs and the characterization of these LNPs, and then we did what we called primary screening. So basically, a primary screening is what we did at the beginning to see if the LNPs that we were doing with these lipids were good, and that meant that normally 80% of the lipids at the beginning were discarded because they wouldn't be good for these primary screening. If the 20% that went beyond that primary screening were submitted to a secondary screening, which is basically a more thorough investigation of how these lipids behave in LNPs. And with that, we normally discarded around 40% of these lipids. And we not only did we do this, what we did was take the data that we got from these primary and secondary screening. We took the knowledge that we got from these two screenings, and we took it back to the system so we could create better and better design of our lipids. So thanks to this approach, we found several families of ionizable lipids with very good results in vivo. You have to remember that all the lipids that we have in stock or in the catalog, they have been submitted to in vivo testing. And the reason for this is that, as you might know, there's not a very good correlation between in vitro and in vivo. So if you want to see small differences between lipids to see which one works best, you normally have to resort to in vivo testing. So we have good results compared to gold standards that are used in the market, like SM102, ARC, or MC3. But obviously, we have well, this is the proprietary structure of one of the lipids that we have in the catalog. As you can see, they have an ionizable polar head, as you would expect. They have aliphatic tails, which in this case are branch tails, and they also include biodegradable groups such as amide and ester bonds. They also include this ester bond is included in what is called a beta thioester, which means that there might be or there is going to be an intermolecular reaction between these thiol group, this thioether group and the ester group, and that means that the biodegradability of this bond is going to be superior than what you would find with a normal ester. And also, one of the things about these lipids is that some of them are already available in GMP. So anybody that wants to go to clinical development, they can already go and have them in GMP quality. So apart from doing RNA with luciferase, we have encapsulated different cargoes. And one of the things that we have encapsulated is SARS-CoV-2 RNA, or RNA encoding for these epitopes. So basically, you can see that we have the lipids in our library. They give a very good antibody response compared to the gold standards that are used in the industry, and is the same for T cell response. In this case, the Th1 was a bit higher than, in some cases, than the one that you find for those two candidates, and the Th2 response, it was very similar to them. We did also challenge study. In this case, it's a bit special, this study, because apart from using one lipid in our library, we also used a formulation that had been optimized in terms of lipid ratios. And also what we did was we used an RNA that had been optimized in terms of UTR and codon optimization, so we could get a better result in terms of antibodies. And as you can see, compared to gold standard, in this case, the gold standard was Pfizer's vaccine. You can see that the antibody expression was a bit higher than the one that we found for the gold standard, and the survival rate was excellent, was what you would expect. So there were not deaths from the challenge study, and the weight loss, it was really similar to the gold standard as well. We have also encapsulated different cargoes. A lot of people are asking us to encapsulate self-amplifying RNA or circular RNA. So when we get encountered by these clients, what we do is we do screening processes into which we encapsulate these cargoes to see which lipid in our library or which formulation could be the best result for them. So in advance to these requests from the clients, we have done this already, and we have some candidates that work at least as good or even better than the gold standards for these two specific cargoes. And the same with CRISPR-Cas technology. In this case, as you know, there's one lipid that is in vogue at the minute, which is LP01. And doing a screening process of our library and doing a screening process of the formulations, we have achieved one lipid that it's much better in terms of gene editing with CRISPR-Cas compared to the gold standard. And also the safety, the in vivo safety analysis of the hepatic enzymes after this injection have shown that there's a good result in terms of safety of this formulation and this lipid. Another cargo that we have used as well with our lipids is the oligonucleotides. So this is something that's not a lot of people are asking, but because oligos, since they are stable by themselves, normally you don't need to include them in an LNP unless you want to take them to somewhere specific. So we have done it anyway, and we have seen that the innovation, the protein expression innovation that we achieve with our lipids and our formulations are very similar to the gold standards. And in this case, what we also did was compared in a study what would happen with decreasing doses of these LNP oligo formulation. And what we saw was that by decreasing the doses, you could see a decrease in the protein expression inhibition for the gold standard, but you couldn't see it for ours. So that's quite a good result. So this is, as I was saying, this is not something that people are asking a lot, but when you want to do some targeting, when you want to take your oligonucleotide to a specific place, that might be something that could be interesting. Of course, as I explained with a CRISPR-Cas slide, we pay a lot of attention as well to the safety profile of these lipids. This is something that is basically part of the screening process. So part of the secondary screening process is looking at the safety profile of these lipids, and that means injecting them in a big dose. That's 50 micrograms of RNA per mouse, or what would be equivalent to 2.5 milligrams per kilogram. And after the injection, we check first the vital constants and how the mice are behaving, but also we check how the hepatic enzymes, some of the hepatic enzymes are altered by this injection, and compared to gold standards, and also to the injection of a buffer of PBS. As you can see, we have several examples of all the examples that we have in the catalog. They have been submitted to this, and you can see that the difference between the first injection, second injection, and the buffer and the gold standards, they're not significant. So we can say that there's no there's a good safety profile in these lipids. Also, we have an NHP study that is ongoing along March. So we should have some results from that by the end of March, beginning of April. And we are quite optimistic that the results are going to be good as well. So I'm going to talk a little bit as well about thermostable LNP formulations. So basically, when we started this pharma business, one of the things that we realized is that during the COVID pandemic, there was a bit of a scare from people about the storage of these kinds of vaccines, these kinds of therapeutics. So we in that company, which was started in 2002, in the other part of the company, we had a lot of experience in terms of stabilizing, or in terms of freeze drying different kinds of molecules, such as antibodies, for example. So that gave us a very good starting point to try to find some conditions that would be good for our LNPs to try to find a method that could give us freeze-dried LNPs that would remain stable and active for long periods of time. So we started from that point, and the stability that we have done after the optimization of this freeze-drying process shows that we have a very good method that can remain, or can keep the lyophilized LNPs active and stable for long periods of time after more than a year of being stored at 4 degrees. That was our objective. So basically, we wanted to be able to keep them in these conditions. And if you follow this kind of method for freeze drying the LNPs, we have seen that we have achieved it. This is basically a similar experiment, but in this case, we overcame what were our expectations. So as I was saying, our objective was to be able to keep these LNPs after they had been freeze-dried in normal fridge, but we also included a group in which we kept these freeze-dried LNPs at room temperature at 25 degrees. And we have seen that the antibody response that we achieve after more than 12 months of storage at room temperature, having been lyophilized, they're very similar to the ones that we achieve at minus 80 degrees in liquid format. Okay, so one of the people, I remember when I started going to conferences, when you talk to people and you told them that you had lipids, so people tend to be really interested, and it was something that gave us a very good selling point. But there was a moment that people started asking for targeting of these LNPs. So we had already started doing this targeting a couple of years ago, because one project that we have in the company, which is for a tuberculosis vaccine, we had done some experiments about delivering these LNPs through intranasal administration. So through intranasal administration, we saw that we could effectively reach the lungs with very good selectivity and with very good protein expression. However, we also started researching the bibliography, and we came across this salt alternative, but by a company that is called Recode. And this company, basically, if somebody doesn't know about it, basically what they do is they change the physical characteristics of the LNP by adding a fifth component to the LNP. So this fifth component would be a charged lipid, a permanently charged lipid. So that would be a lipid that is permanently positive or permanently negative. So the adding of this compound to the LNP, what it does is going to change the physical characteristics of the LNP, is going to change the charge of the LNP, and supposedly that's going to produce some targeting in the LNPs, because the protein corona that is going to get surrounded, or it's going to surround this LNP in the blood current, is going to change, and that protein corona is the one that is supposed to give this passive targeting to a different organ. So we tried to, what we did was we took some of the lipids in our library, which were ionizable, and we made them permanently cationic. So basically, with one reaction, one step reaction, what we could is we could methylate the amine group, and that methylation made them permanently cationic. And what we did was include them in the formulation. To our surprise, so the selectivity that we achieve with the combination of our ionizable lipid and the permanently cationic lipid that came from that ionizable lipid, so it gave very similar, a little bit more selectivity to the lungs than the one that we had achieved with Recode salt, which was MC3 and DOTA. But the good thing about us is that the protein expression that we achieved was higher than the one that you could achieve with the Recode alternative. We hypothesized, and there's a paper that we wrote about it when we talked about our lipids, we hypothesized that the reason for this was that we used the ionizable lipid and the cationic counterpart of these ionizable lipid, and that meant that the packing, since they were basically the same lipid, the packing of the LNP was better, and maybe that made the LNP work better, but we don't know about it. Anyway, when we had done this, one of the things that we thought was, okay, so we have in our library, we have a very good screening process, a very huge screening process that we have done, and in which there's some lipids that we have discarded because the LNPs that we achieved with those lipids gave us some physical characteristics that went beyond what was reported to be good for ionizable lipids. So what we did was we went back to the screening process, to our screening process that had been intramuscular, and what we did was we took back some of the results that we hadn't injected because we thought that they were not going to give a very good result intramuscularly. So we took these lipids And basically, we injected them intravenously. And to our surprise, we found that some of the lipids in our library, in a normal composition, that means ionizable lipid, cholesterol helper, and the pegylated lipid, would give us a very good selectivity to different organs, which would be the lungs or the spleen or a combination of both, with very good protein expression. So as you can guess, what we did, we took these the structure, we started analyzing this structure that gave us these very good selectivity, and what we realized was that there was a common structure within our library that seemed to have a very good targeting and specificity for the lungs or the spleen, and with very good protein expression. So as you can guess, we started making combinations. We started making adjustments of these lipids, and thanks to this, we found some examples in our library that would give us very good protein expressions and selectivity for the lungs and the spleen. In the case of the spleen, which you can see on the right, you can see that there's one Certest spleen 1 that has been injected as well, has been analyzed with something that is called Certest sec plus spleen 1, which is the first bar on your left. So the reason for that is that in this case, we did a twist to the analysis, because apart from using a formulation with one of our lipids that gave spleen targeting, we also included an RNA sequence that gave very good protein expression or very good selectivity in the spleen. So we thought that the combination of both the RNA sequence and the spleen targeting LNP would give a better combination than if we used another kind of RNA. As you can see, the selectivity is pretty similar to the one that we found for the second case, but the protein expression that we achieved was much higher. So we have performed some cytometry analysis, and we are still performing this cytometry analysis. But the first initial cytometry analysis of these spleen targeting LNPs show that we can reach different cells within the spleen, and some of the cells that we reach are the T cells, which might be important for technologies such as the CAR T technology. So this would be a comparison between what we achieve in the lungs compared to the sort alternative. So as you can see, the first on the second on the right would be the record sort, which would be MC3 plus DOTA. The second bar, which would be a combination between our fifth cationic lipid and the ionizable lipid, which has very good selectivity but improved expression respected to the other one. And in the first case, you can see what we can achieve with the Certest 4-component LNP that is targeting the lungs, which has very good selectivity and also very good protein expression. One of the things that we did as well is to include these formulations that we have seen in the previous slide, but using small RNAs to see if we could affect the same or have the same effect, but with a different kind of cargo. But in this case as well, these experiments served us to explain some of the doubts that we had about these LNP, or about the targeting of these LNP. Because one of the questions that we had is, we didn't know if what was happening is that the LNP was reaching, for example, the liver in this case, but it wasn't expressing the liver, so it was kind of switched off in the liver. Or if what was actually happening was that the LNP was not reaching the liver, and most of it was reaching the lungs. So what we did was we did qPCR to analyze this, and what we found is that with the Certest lung approach, what we found was that most of the RNA was found in the lungs. So that would explain that it was not reaching any other organs. It was mainly reaching the lungs. So it wasn't being switched off in the rest of the organs. It was just basically reaching the lungs as the main organ. So, of course, one of the things that one realizes when we're talking about passive targeting is that passive targeting is kind of limited. So with passive targeting, if you have a normal LNP, you normally reach the liver. If you do something specific to the LNP, as we have seen, you can reach the spleen or the lungs. But if you want to reach other kinds of organs, other kinds of cells, you have to start doing something different. And that something different is active targeting. So active targeting, what we call active targeting, is including some ligands on the surface of the LNP that we think or that we have established that might have some affinity for a specific receptor in a specific organ or cell type. So basically, what we do is we include peptides or antibodies on the surface of the LNP, and with this, we try to take the LNPs to a different organ or a different cell type. So we do have some data already, but basically the way in which we do this is we identify a target. So basically, we identify a receptor that might be of interest for a specific organ. And in the case of peptides, we design the peptide sequence that can have affinity for this receptor. We design this peptide. We make the synthesis of the peptides, which is something that we do thanks to a peptide synthesizer that we have acquired, and with which we can do more than almost 50 sequences, peptide sequences at the same time. So basically, we do small modifications in the peptide design because maybe you don't want that peptide to have so much affinity to the receptor, or maybe you want to improve the affinity that it has with that receptor. So you have to tweak a little bit that peptide sequence to be able to reach the one that works best with your LNP and with your solution. We do the conjugation of these peptide to the LNP or to the oligo, and then we do the in vitro and in vivo evaluation to see which of these alternatives would work better. So this is an example of the optimization that we have done. So basically, we use click chemistry for these conjugation. In this case, you have to twitch a little bit the formulation as well, because obviously you're including different kinds of chemistries and different kinds of substituents to the surface of the LNP. So that might change your LNP characteristics. So we have had to make some adjustments to the LNP and also to the kind of conjugation that you can do to be able to include this conjugation or this ligand on the surface of the LNP. But basically, what we do is we have the pegylated lipid with one end, with the end that has been or that includes a reagent that is that can react with the other part of what is going to be the therapy, which is basically the peptide, and we have optimized this conjugation to make it work better. This is some of the results that we have achieved with this. And in this case, what I'm showing here is the comparison between two different cell lines and two different conditions. So the two different cell lines, we have one cell line that has a high affinity for a kind of receptor or a kind of peptide, which is the peptide EGFR, and another one that has some affinity, but it's not as high as the first one. The LNP has been, in the first case, it has been conjugated with this EGFR peptide, and the second example is just the LNP by itself. So in the first case, in the top side, you can see that when we include the peptide on the surface of the LNP, the uptake of this cell type is much higher than the one that you achieve with without the peptide on the surface of the LNP. But also, if you compare between those kind of those two cell lines, you can see that the percentage of transfected cells that you achieve with the one on top, with the one that has affinity for that peptide sequence, it's much better than the one that we achieve with the other one, which makes or kinds of explains or kinds of verifies that the peptide sequence, or that the way that the uptake is being done is by the affinity with of this peptide with the receptor. So it's kind of specific to this peptide sequence. Similarly, to prove this as well, what we did too was to compare how the uptake of these LNPs would change if we pre-incubated the cell lines with the peptide sequence, to see if this was the way in which these LNPs were being uptaken. And as you can see, there's a big difference between the peptides or the LNPs that have a peptide on the surface, the uptake that you achieve on the cell line, and when you don't have that peptide on the surface. But also there's a big difference between what you achieve when you pre-incubate this peptide with a cell line. In this case, you can see that the uptake is much smaller, and that's because what we theorize is that this uptake or these peptide sequences specific is what is making this uptake by the cell line to happen. And similarly, this is what we achieve with the oligos. So this would be conjugation of oligos to similarly to the LNPs. So the process is very similar, and the result that we achieve is very similar. In the ones that we have the peptide, the uptake is much better than the ones that don't have the peptide, but also it shows there is a specific, because the cell lines that have these affinity for this peptide work much better in the uptake than the ones that don't have this high affinity. So just to wrap it up, so this would be the way in which we do partners, or we do partnership with different companies. So basically, we like to say that we are very flexible in how we do business, and I think we are. So basically, what we do when we have interest from a company in our technology, in our lipids, for example, we're open to almost any kind of collaboration. So there's some companies that they come to us and they ask us, okay, so I have a cargo, but I don't want to send you my cargo. So maybe you can send me the lipids, I try them. We can sign an MTA, we try them, we see if we like them, and then we can talk about how we license this. We're happy with that, but there's some other companies that they don't have the ability of doing an LNP, of encapsulating. So we can do that as well. We can do it as a service. So they send us the cargo, we do the screening, or we do the encapsulation into LNPs. We send them back, they try them, and the process from that is similar to the first case. So we work normally with MTAs. Then once we have interest from a company, we sign a license. So we sign a license, which is basically an interest from the company. And what we like to say is that we try to help companies in every kind of need that they might have. So similarly to conjugation to the formulation into LNPs, we can also help them with the evaluation in vitro or in vivo, or if the project is a bit bigger, for example, they want a specific targeting to a specific organ, we can also help them in the ligand selection, so peptides or antibodies. We can help them in the conjugation of these ligands. We can help them in the evaluation. It's basically up to the client. So I think that's me for today. Once again, it was a pleasure to be here. It was a pleasure to talk to you. And if you have any questions, I don't know if we have time for that, but I'll try to answer them.
Thank you, Javier. Let me see if I can switch to the next slide or not. Yes. So as a key takeaway, we would like to emphasize that the collaboration between TB Bio and Certest Pharma will enable a seamless transition from discovery to in vivo delivery. And as said, to wrap up this session, let's move on to the Q&A session. This is your opportunity to ask any question and get insights directly from our experts. So feel free to share your thoughts. I'm here with Erika, just next to me. So if you have any question about in vitro delivery or analysis, excuse me, let me know, let us know. And so about the question, so I have already, just 1 second, I need to stop sharing to see the questions. Okay, so we have few here. So those, the same lipids perform the best on mRNA and saRNA.
No, so basically when we do these slides, we put names like Certest Lipid 1, Certest Lipid 2, but they're not exactly the same. So one of the things that we have to do when we encounter a different cargo, even if it's the same, for example, it could be that a lipid works best for RNA with luciferase, but then we go to, for example, RNA that is going to go against, I don't know, I'm going to make it up, malaria for a virus, for example, for antibody expression, and that's not the one, or the one that gives you the best RNA luciferase expression is not going to be the best one for these kind of therapy. So what we normally encourage people to do is we encourage them to do a small screening process. So it could be, I don't know, normally we go from 3 to 5 different lipids to see which one would be best for their application. That's normally what we do. And we do it with different cargoes, but also within the same cargo, we normally encourage them to try more than one, because sometimes there can be small differences and it can be worth exploring.
Thank you. Here's another one. Is the freeze drying applicable to any type of LMPs or is associated to specific ionizable lipid?
No, in fact, the first LMP optimization of freeze drying optimization that we did it with SM102. So basically, the optimization of these LMP freeze drying was through optimizing the timings for the primary and secondary drying, and that's basically what you need to optimize. It's a bit more complicated than just letting it in the machine, letting it dry up. It's you have to tweak a little bit the parameters, but what we have seen is that once you find the formulation, not the formulation, but the method for freeze drying these LMPs, it's quite robust. So you normally can use it with different kinds of LMPs, with different kinds of ionizable lipids.
Thank you. What is your specific RNA sequence that improve the transfection?
I'm guessing that they're talking about the spleen. So basically what we did, or it could be also with the first case with RNA luciferase. So what we realized was that the RNA, the UTRs and current optimization that we used, it was giving us a very good result, and that's what we filed a patent for. I think that they're referring to that. So basically, we have a patent for this optimization of the UTRs. So we have seen that they work very well. We have a partner that I think I cannot disclose at the moment, that they're really interested in licensing this technology, because not only in our hands but also in their hands, they have seen that there's quite a difference between the protein expression that you achieve with these UTR optimization and the normal UTRs that people tend to use. So, yeah.
Thank you. I've asked if the person wants to specify.
Okay.
Knowing the diversity in cells types in lungs, do you know which cells are specifically targeted with your lung-targeted LNPs?
So that's something that we are doing at the moment. The reason we haven't done it yet is because we didn't have the mouse model that was needed for this. So we are working with a university. They're the ones that let us use their facilities to do the experiment. So all the experiments that we do, I think there's a note in one of the slides, but all the experiments that we do, they have been approved by ethics committee. So we like to do things by the book, and that's how we do it. But we didn't have a model for being able to have very good sensitivity for the cytometry analysis. So that's something that we applied for a few months ago. It was approved not a month ago, a bit less than that. And we have recently got the mice here to do the cytometry analysis. So we should have some data soon about the kind of cell types that we can reach with the different targeting applications.
Thank you. Did you try to combine lung targeting LNP within an aerosolized formulation? If
Actually,
yes,
yes, actually, yes. So as I was saying, the reason why we start exploring the inhaled administration was because it was related to this tuberculosis program. And our idea was to combine freeze drying, inhaled, and lung targeting to these tuberculosis program. So we have actually tried some aerosol devices. The problem with that is aerosol devices, they have to be very specific for the LNPs because otherwise they tend to break the LNPs. We have, however, talked to one company that they have a technology that is kind of different to the kind of technology that uses aerosol as just particulates that are made smaller. So we are exploring that option. We have talked to this company. They're going to try to see that the LNPs remain stable in terms of physical characteristics after they have applied their technology. And we are very optimistic in that. So that's something, it's a very good question because that's something that could actually even change the cell type that you can target with a route of administration. Because sometimes if you go intravenously, the cell types that you can reach are different to the ones that you can reach inhaled. So it's something that we're definitely going to explore. And this company is going to help us do it.
thank you. So we have still a few minutes. I think we have 3 or 4 more. Do you have any lipids or formulations that are suited for certain delivery routes, for example, subcutaneous? And if not, would it be possible to target screening towards this based on the lipid descriptors?
So we have actually tried microneedles, which would be good for subcutaneous administration. In fact, one of our partners is going to use microneedles in their application for a vaccine. I guess what they're trying to do, I don't know the type that they want to, the type of cell that they want to reach, but that's how they have seen that they can affect the most antibody expression. So we are actually not directly us, but we have some partners that are using these kinds of administrations with their technology. So we should have some data in a few months that we can share. Yeah.
Okay. So the next one, the Certest sort LNP looks to have lower overall delivery efficiency than the gold standard LNP. Any explanation for that?
Well, to be honest, that's something that doesn't really worry us because that's not the alternative that we want to use. I mean, the reason I normally talk about this is because that is what gave us the idea to start the screening process of our own long targeting or passive targeting approach. So I normally use that as an introduction of how we came across the idea of trying to screen our LMPs. But it's not something, I wouldn't have an answer to that. I mean, I don't know why that is the case, but it's not something that's really worry us because that's not the alternative that we want to use. We wanted to be able to have something that would give us a similar result, or if possible better, by using a normal composition, a full component composition. So that was our business, if you want to call it like that.
Okay, thank you. Do you have still tested LNP formulations for the selective delivery of RNA or DNA in cardiac tissue?
In cardiac tissue, that's something that a lot of people are asking for. So basically, people are asking for kidneys. They're asking for, obviously, for crossing the BBB. They're asking for the heart as well. So that's something that we don't have at the minute, but it's something that we are working on. And that's something that we are going to have to do with active targeting. So the problem with active targeting is that the possibilities are endless. So we are a company that we don't do things because we want to do them. We do things because people ask us to do something, or because we have identified that the clients are interested in something. So, for example, in terms of passive targeting, we started because we realized that there was some interest in that. And active targeting is something that we have identified because a lot of people are asking about it as well. The problem is having an alternative ready for any kind of cell type is kind of difficult, as you can guess. So we try to move in a case by case. So we can help these companies develop an alternative that can take their LNP to the place, to the organ that they want to reach. So we can actually put in place our conjugation technology, our LNPs, and the peptide that we can identify to take it to these places, or the antibody that we can conjugate as well.
Okay. Two more. Can you tell us more about the routes of administration tested of toxicity?
Yeah.
For
So for toxicity, is intravenous. So basically, we want to be in the worst case scenario. So first of all, you have to do it intravenously, because that's the worst case scenario. So basically, injecting directly into the blood torrent, what's gonna give you the best idea of how toxic or how non-toxic your lipids and your formulations are. But also, it's a question of volume. If you try to inject, I'm not an expert on animal handling, but basically, I think that if you try to inject 50 micrograms, which is quite a big dose of LNP formulation, it wouldn't fit. I mean, you wouldn't be able to put so much into the muscle. So it's a question of, we don't have another option. We have to do it intravenously, but also because intravenously is what is going to give you the best idea in terms of toxicity. So that's the route of administration for that.
Thank you. And last one, for spleen delivery, can you determine approximately how many percentage of T cells were expressing?
So same again with the lung targeting. In terms of spleen targeting, we are still waiting for some results. So we got some preliminary results that they're telling us that we can reach the T cells, but we have to take them with a pinch of salt, because basically what we saw was that the sensitivity was a bit low with GFP. So we are waiting for some new results with this animal model that will give us a better understanding of how much T cells, B cells, macrophages we're reaching in the spleen. So that's something that I cannot answer at the moment, but probably in a month, more or less, we should have some beautiful data about it.
So we need to keep in touch then.
Yeah.
All right. So I think that, well, I don't see anything more. Let me see. Yes, there is another one that just came up. So, hello, is there any forecast for the in vivo studies using the peptide conjugated LNPs?
So for peptide, yes, well, we try to move very fast with this, and I think that we do have some preliminary data as well from some studies that we did on mice, in which we planted a tumor, because this EGFR is supposed to be targeting tumors. And we have some preliminary data that are telling us that we can reach the tumor, but it's something that before we can just I don't know, just shout it out. We want to double check. So it's something that basically in a couple of weeks we should have some more or less definite data about it. But it was looking promising. I can say anything else, but it was looking good.
Well, thank you again. Then before we conclude, I'd like to ask to take a few moments to complete the survey that will appear in your browser when you leave the session. And otherwise, again, thank you very much for all your attention for joining today's webinar. And we hope the information shared has been really helpful and inspiring. Thank you for this session. And yeah, feel free to reach out if you have any other questions, and if you have others that come up afterwards, there's no issue. We'll be glad to get back to you. So thank you, Javier. Thank you, Laura. Thank you, Erica, next to me. And have a good rest of your day. And
Okay,
good week
you
as well.
too.
Thank
Okay.
you.
Okay, see you
Bye-bye.
later. Bye-bye.